Hello 11 advanced students. Today we complete the second lesson that we've got, and today we're going to talk about the Kelvin cycle. Right, as usual, let's start with an overview of the Kelvin cycle, which is also the light independent reaction. So let's have a quick overview of what we have. So phase 2, which is the light independent reaction or the Kelvin cycle. The Kelvin cycle. Where does the Kelvin cycle take place? Well, if we remember from the overview of this whole lesson, from overview of photosynthesis, we know that it takes place in the stroma of the chloroplast. Where do the ATP and NADH come from that we use? They come from phase 1. From the light independent reaction. And what is the final product of the Kelvin cycle? Glucose. Okay, let's have a more in-depth overview. So we said that the site, it occurs in the stroma of the chloroplast. For this process, we do not need light. And that is why it is called light independent reaction, because I don't need light. ما بحتاج الضوء. تمام؟ Raw material, material that we're using, or the material that we're using today, in the cycle, CO2, ATP, NADPH, and the product is, as we said, we have glucose, but we'll also see the other products that are generated in this cycle. So let us start. Okay, so although NADPH and ATP provide the cell with large amounts of energy, these molecules are not stable enough to store chemicals for long periods of time. So NADPH and ATP provide the cell with large amount of energy, but they cannot stay stable. They cannot store energy and stay stable for a long period of time. Thus, there's a second phase of photosynthesis. ليش أنا عندي second phase? Why do I have the second phase of photosynthesis, which is called the Kelvin cycle? Because I need to change NADPH and ATP into a more stable form. هلا Kelvin cycle is in which the energy is stored in organic molecules such as glucose. So we store the energy in organic molecules like glucose. Okay, so as we know, it's also referred to as the light independent reaction. So let's follow along to look at the steps of the Kelvin cycle. Right, so the first step of the Kelvin cycle is called carbon fixation. So step 1 is called carbon fixation. Carbon fixation. In this step, we have 6 carbon dioxide, so 6 CO2, 6 carbon dioxide, enter the cycle from the atmosphere. Now, the carbon dioxide molecules will combine with 6 5-carbon compounds. So it will combine with 6 5-carbon compounds to form 12 3-carbon molecules called 3-phosphoglycerate or 3-PGA. This process is called carbon fixation. The joining of carbon dioxide with other organic molecules is called carbon fixation. Now, it's important to note, this isn't written in the book. هاي معلومة مو مكتوبة بالكتاب. So just focus with me. If we go back to... Okay, there is a mistake in this. The Rubisco... Okay, the Rubisco is supposed to be here. I think this is like, there's something wrong with the PDF. Tamam? So the Rubisco is supposed to be here. There's no Rubisco here. Okay, let me get rid of that. there is no Rubisco here, no such thing. We don't have any Rubisco over here. Okay, what we do have is the Rubisco up here. So what is the step that they didn't tell me about? They said the 6 carbon dioxide will combine with 6 5 carbon compounds. What are these 5 carbon compounds? They just said 6 5 carbon compounds. إيش هم؟ Actually, what happens is the Calvin cycle produces these 5 carbon compounds اللي هم إيش؟ اللي هم RUBP. So at the end of the Calvin cycle, we have Rubisco, and the Rubisco creates the 5 carbon molecule called Rubulus 1,5 biphosphate, which is RUBP. So the 5 carbon compound that the CO2 is combining with is RUBP. And what happens is إنه الرubisco مش بس بيساعد بعملية إنه إحنا نعمل ال RUBP، ولكن أيضا بيساعد على الفيكسيشن، على combining of ال CO2 and the RUBP. So the Rubisco doesn't just create the RUBP, but it also is the enzyme that helps combine the 6 carbon dioxide with the 6 RUBP. 6 carbon dioxide و 6 RUBP. ال RUBP شو هو؟ It's a 5 carbon compound لأنه فيه 5 carbons 1 2 3 4 5 و 6 CO2. تمام؟ They combined, and this combining is called carbon fixation. طيب, when we combined 6 CO2, 6 RUBP, the product is 12, so this is how many? 12 of the 3GA, and 3GA is a 3 carbon molecule, and it is called 3-phosphoglycerate, 3GA. It is a 3 carbon molecule, لأنه فيه 3 carbons. It is called 3-phosphoglycerate, 3RGA. This first part is called carbon fixation. This first part is called carbon fixation. So step 1, another name for step 1, الاسم الثاني للخطوة الأولى هي carbon fixation. تمام. الخطوة الثانية, the second step that we have is called the reduction phase. Reduction. الاسم الثاني إلها هو reduction. هلا in the second step of the Calvin cycle called reduction, تمام, the energy stored in ATP and NAD pH is transferred to the 3-phosphoglycerate. So الانرجي اللي موجودة في ATP وفي NAD pH تتحول بعطوها they transfer it مش بحولوها they transfer it بعطوها لل 3GA molecule. تمام. to form high energy molecules called شو ايش هم هدول ال high energy molecules? They are called glycerate 3-phosphates G3P. تمام. So the ATP will NAD pH transfer their energy لل 3GA لحتى يعملوا so that they form a high energy molecule called G3P اللي هو glycerate 3-phosphate. Okay. So let's look at this here. So what happens is in this process ل 12 ATP حيكون عنا 12 ATP. Okay, we will have 12 ATP. Okay, what the ATP will do is it will give one phosphate group. تمام. So ATP will lose the phosphate group. Once it loses the phosphate group, what will happen to ATP? It will turn into ADP. So عندنا هنا to the 3 PGA. حنزيد شو؟ we're gonna transfer the energy from ال ATP وال NADPH. كيف؟ we'll have 12 ATP. They will lose 1 phosphate and they will give it to the 3 PGA. فحيكون عندي 12 ATP بتتحول ل 12 ADP لأنه كلهم خسروا الفوسفيت تبعتهم وعطوها لل 3 PGA. بعدين ال NADH سوت نفس الاشي. we have 12 NADPH. Well, NADPH gives what? It gives up a hydrogen molecule and a hydrogen ion and an electron. So the NADPH will lose a hydrogen ion. It will lose a hydrogen ion. هتخسر hydrogen ion وحتخسر electron وحتعطيهم لل 3 PGA. فحيتحولوا ال NADPH إلى إيش؟ إلى NADP+. حيرجعوا زي ما كانوا قبل ما زمان، يعني إذا بتتذكروا electron transport chain. تمام؟ Okay, so what happens in the second step, which is the reduction step? The 12 ATP will reduce into 12 ADP because it will give away its phosphate to the 3 PGA. And then the 12 NADPH will reduce to 12 NADP plus because the NADH will lose hydrogen ions and electrons and it will give it to the 3 PGA. This step is called what? Reduction. What's the product of reduction? The product of reduction is 12 molecules of the G3P. Let's not forget G3P is a high energy molecule called glycerhydrate 3-phosphate or G3P. تمام? Okay. Now the third step. In the third step, okay, focus here. So in the third step, 2 G3P molecules leave the cycle. They leave the cycle and they are used for the production of glucose and other organic compounds. So 2 of the 12 G3P that I had from step 2, 2 of them will be removed from the cycle. They will leave the cycle. And the plant will use these 2 molecules to create sugars with starch, amino acids, lipids, and other compounds that are needed for the plant metabolism and structure. Tamam? The remaining 10 are converted into 6 molecules of ribulose 5-phosphate اللي هي regeneration step. So هنا عندنا the 12 G3P that I have, the 12 G3P, 2 of the 12 G3P will be transported from the chloroplast to make glucose with fructose, with starch, with lipids, all the other things that the plant needs. تمام? So this is in step in the reduction step. تمام? So 2 of these G3P will leave. So now I am left with 10 G3P in the cycle. These 10 G3P will be used to create RUBP. كيف? In the final step, the energy stored in ATP converts the 5 carbon molecules to 6 molecules of ribulose 1,5 biophosphate. Tamam? So the remaining 10 molecules, okay? The remaining 10 molecules okay, so this step, all right, one second. So this was not explained in the book, I will explain it. So as you can see here, the 10G3P will be converted into 6 molecules of the ribulose 5-phosphate. Focus on the words. So ribulose 5-phosphate. 5-phosphate. Ribulose 5-phosphate. So the 10G3P will be changed into 6 ribulose 5-phosphate. Okay? Right, so then in the final step, the energy stored in ATP will convert these 6 5-carbon molecules that are the 6 ribulose 5-phosphate. It will change them into ribulose 1,5-biphosphate. And these molecules will combine with the 6 carbon dioxides through the help of the Rubisco enzyme. So the Rubisco enzyme converts the 10G3P. So the Rubisco will convert the 10G3P into the ribulose 5-phosphate. It will change it into the ribulose 5-phosphate, and then with the power of 6 ATP, it will change into the 6 ribulose 1-biphosphate. So the Rubisco, which is an enzyme, will convert the 10 remaining G3Ps into the 5 molecules of RUBP. So the 10 G3Ps will be changed into 6 RUBP by the help of Rubisco. So yes, this is an old book, old PDF of the book. So there is Rubisco here, but in our book it's not there. So just look at the image in the book, but just know this, that the 10G3P was changed into 6 ribulose 5-phosphate first, and then with the help of 6 ATP, the energy used from them, it was changed into 6 ribulose biphosphate by the help of the enzyme Rubisco. Okay, so the 10G3P was changed into this by the help of the Rubisco enzyme. Tamam? And the Rubisco enzyme is also the enzyme that helps these 6 RUBP fixate with the carbon dioxide. So Rubisco is a biological enzyme, and it converts inorganic compounds, inorganic carbon dioxide molecules into organic molecules that can be used by the cell. So because Rubisco, let's look at this in the book. So because Rubisco converts inorganic carbon dioxide molecules into organic molecules that can be used by the cell, it is considered one of the most important biological enzymes. And the plants use the sugars formed during the Calvin cycle both as a source of energy and as building blocks for complex carbohydrates, including cellulose, which provides structural support for the plant. So to just go over the steps really quickly, and as I said, the last step is called regeneration. Why do you think it's called regeneration? Because I used RUBP in the beginning of the cycle, so I need to regenerate it. I need to remake RUBP. So in the first step of the cycle, called carbon fixation, I have 6 CO2 molecules and 6 5-carbon molecules, which are called ribulose 1,5-biphosphate, or RUBP. Rubisco, which is an enzyme, will help fixate the 6 carbon dioxide to the 6 RUBP. This will result in 12 3-PGA or 3-phosphoglycerate. We'll have 12 of the 3-phosphoglycerates. This is the first step, called what? Called carbon fixation. In the second step, called reduction, what we'll have is we'll have 12 ATP lose their phosphate and give it to the 3-GA, and therefore by losing one phosphate it will become ADP, and the NADPH will lose a hydrogen ion and will lose an electron, and it will give it to the 3 PGA, and in this process it will return back to NADP+. The product of step 2 is 12 G3P, which is a high functioning or a high energy molecule. Then in the last step, which is called regeneration, from the 12 G3P that we created, or that we have, 2 will leave the process and they will be transported to the chloroplast to make glucose, fructose, starch, etc. The 10 G3P that are left are converted into 6-ribulose 5-phosphate, 5-phosphate, not just phosphate, into 6-ribulose 5-phosphate with the help of the Rubisco. These 10 G3P will be changed into 6-RUBP. How? They will first be changed from the 10 G3P into the 6-ribulose 5-phosphate, and then with the energy from 6 ATP, it will be changed into 6-ribulose 1,5 biphosphate, which is RUBP. Okay, 6-RUBP. So these 10 G3P will be changed into 6-RUBP, but don't forget that they first change into the 6-ribulose 5-phosphate, and then with the help of ATP and the Rubisco, they are transformed into the 6-RUBP, which is ribulose 1,5 biphosphate. And that is it for the Calvin cycle. I hope the process makes a bit more sense for you now.